Okay, so welcome back to this next video in which we are discussing drug addiction. Okay, so we are in the process of discussing many different examples of addictive drugs, and we're looking at how these addictive drugs all raise dopaminergic transmission within the nucleus accumbens and the medial prefrontal cortex. Okay, so the next example of an addictive drug that we are going to talk about is nicotine, okay, which is the active ingredient of tobacco. Right, so nicotine is going to work by acting on nicotinic acetylcholine receptors. So I firstly want to start off with a discussion of the nicotinic acetylcholine receptors. So for short, the nicotinic acetylcholine receptors are often written as the NACHRs, like so. Okay, the little N stands for nicotinic. Okay, the ACH stands for acetyl. Okay, so let me just colour this in, colour code it. So the little N here is for nicotinic. The A is for acetyl. Okay, then the CH is for choline. Okay, so that's that portion, nicotinic acetylcholine, and then the R is for receptors. Okay, so nicotinic acetylcholine receptors. Right, so let's discuss the structure then of nicotinic acetylcholine receptors, but I think probably before we discuss the structure we should discuss what is the ligand for all nicotinic acetylcholine receptors. Well, the ligand for acetylcholine receptors is of course acetylcholine, okay, which is an incredibly important neurotransmitter in the peripheral nervous system, okay, but although it does have functions in the brain as well, okay, its functions in the brain are rarer than its functions at peripheral sites, okay, it's famous for working in the neuromuscular junction. Okay, so let's discuss then the structure of acetylcholine. Okay, so basically it is an ester formed between acetic acid and choline, which is an alcohol. Okay, so let me show you its structure. So here is the acetyl group here, okay, which is the acyl group corresponding to an acetic acid molecule. Okay, like so. And then it will be attached via ester link to a choline alcohol molecule. Okay, so the choline molecule consists of an alcohol group which is now bound to the acetyl group here. So let's highlight this in. So this portion here, okay, this that I'm highlighting in orange here, this is the acetyl group. Okay, and everything that we're now showing is going to be the choline molecule. Okay, so here's an alcohol group that is no longer an alcohol group because it's bound in this ester link here. Okay, then we've got the ethylene group here, and then on the end we're going to have a nitrogen atom which is going to have 3 bonds coming off it to methyl groups. Okay, like so. Now, nitrogen is not supposed to have 4 bonds in this way, it's supposed to have 3 bonds. Okay, when it forms 4 bonds in this way, one of these bonds is going to be formed by the nitrogen donating both electrons into that bond, okay, from its lone pair. Okay, so it effectively gives an electron away to one of these carbons and ends up with a positive charge. Now you might think, well, does that mean that the carbon is going to have a negative charge? Well, the answer is no, because when the carbon came into this sordid arrangement, it will have originally had a positive charge. Okay, so to make this clearer, in a covalent bond, okay, like we've got here between this carbon and this sorry, it's the wrong way around. Oh, I'll just say it's this bottom bond rather than, I was talking about this top bond until I drew it the wrong way around. Okay, so we'll say we're now working on this bottom bond here. Okay, in a covalent bond there are 2 electrons. Now normally 1 electron comes from each of the members of the bond, so normally what should happen is the nitrogen will put in an electron and the carbon will put in an electron. In reality, into one of these 4 bonds here, the nitrogen has put both electrons. So let's say it is this bottom one here where it's occurred. Okay, and the nitrogen has given away 2 electrons, okay, into this double bond. Now, because the normal understanding of a covalent bond is that 1 electron belongs to each member, effectively it's as though one of these electrons has now been donated to the carbon atom. Okay, so that means that the nitrogen has lost an electron and therefore gains a positive charge. And you might ask, well, okay, the carbon has now gained an electron, so surely it should have a negative charge, but the reality is that when this carbon came into this arrangement, it would have originally had a positive charge, so when it gained the electron, it's now become neutral, and effectively it's deferred the positive charge onto the nitrogen. Okay, so overall the acetylcholine molecule does have a positive charge. Okay, so this is the choline portion, all of this here. Okay, right, so that's the neurotransmitter that works on nicotinic acetylcholine receptors. Now, nicotinic acetylcholine receptors are all going to be ligand-gated ion channels. Okay, and the shorthand for ligand-gated ion channel is LGIC. Okay, ligand-gated ion channel. And specifically, they're going to be within this superfamily of cis-loop ligand-gated ion channels. Okay, now when acetylcholine binds to nicotinic acetylcholine receptors, it causes the ligand-gated ion channel to open. Okay, it will open an ion channel down the middle, and that's going to allow sodium ions into the cell. So these are also excitatory ligand-gated ion channels, like glutamate receptors, rather than like GABA receptors. Okay, right. So let's now have a look at the structure of nicotinic acetylcholine receptors. Well, the good news is they are very similar to GABA receptors. Okay, so here is the ligand-gated ion channel sitting in the membrane like so. Okay, with the pore going down the middle. Okay, and again, nicotinic acetylcholine receptors are not just one protein. Instead, they are pentamers. They're made up of 5 separate subunits, okay? And if we were to pull one of the subunits of a nicotinic acetylcholine receptor out, okay, so let's pull out this subunit here and have a look at it, we would find that it looks exactly the same as that picture that we drew for GABA receptors. Okay, so let's just draw this again anyway. So the single subunit now, here's the amino terminus, we'll have a cis loop extracellularly here. We'll then have the 4 membrane spanning alpha helices with this intracellular loop between M3 and M4. Okay, so let's colour this in. So we've got the cis loop structure again here. Okay, we've got the 4 membrane spanning alpha helices M1, M2, M3, and M4. And we've also got the intracellular loop between M3 and M4. Okay, so that's why the nicotinic acetylcholine receptors are also counted as being cis-loop ligand-gated ion channels, because they too have this basic structure where the entire ligand-gated ion channel is a pentamer, okay, and each one of the subunits of the pentamer has this basic structure here, where you have the cis loop and this same 4 membrane spanning alpha helices with the intracellular loop. Okay, so that's the running theme between all cis loop ligand-gated ion channels. Now, obviously the subunits of nicotinic acetylcholine receptors are different from the subunits of the GABA receptors, otherwise they would be the same receptors. Okay, so what are the nicotinic acetylcholine receptor subunits then? Well, again, like the GABA receptors, there is not just one nicotinic acetylcholine receptor subunit, okay? There are loads of the things, okay? There are 16 different subunits for nicotinic acetylcholine receptors, as far as I'm aware, okay? And again, they are grouped into families. Okay, so there is the alpha family, firstly. So let's go through the alpha family. So there are 9 members of the alpha family. Okay, and there is one blip in the naming system here, so I'll tell you about it. So there is the alpha 1 nicotinic acetylcholine receptor subunit. There is the alpha 2 nicotinic acetylcholine receptor subunit, alpha 3, and we go on. Okay, all the way up to alpha 10. Okay, well that doesn't make sense. We were supposed to have 9. Okay, well one of them doesn't exist. Alpha 8 doesn't exist in humans. Okay, so no alpha 8 in humans. Okay, so that means that there are only actually 9 alpha subunits of nicotinic acetylcholine receptors. Okay, because there is no alpha 8 in humans anyway. Right, okay, so those are the alpha subunits of nicotinic acetylcholine receptors. That's 9 out of the 16. Let's now go on to the beta family of nicotinic acetylcholine receptor subunits. So there is the beta-1 nicotinic acetylcholine receptor subunit, there is the beta-2 nicotinic acetylcholine receptor subunit, beta-3, and also beta-4. That takes us up to 13 total nicotinic acetylcholine receptor subunits. Okay. Finally, there are 3 that then don't fit into families, or rather they're in families but on their own. Okay, and these are the gamma nicotinic acetylcholine receptor subunit, the delta nicotinic acetylcholine receptor subunit, and the epsilon nicotinic acetylcholine receptor subunit. And overall, that gives us 16 different nicotinic acetylcholine receptor subunits. Now note that even though some of these are called the same thing as the GABA A receptor subunits, they are not the same protein. So for instance, the alpha-1 nicotinic acetylcholine receptor subunit is very different from the alpha-1 GABA A receptor subunit. Okay, so do not let that confuse you. These are different proteins coded by different genes with different primary sequences of amino acids. Okay, they just happen to have these structural features in common once they've actually folded up. Okay, right. So, what I now want to tell you about is the different types of nicotinic acetylcholine receptor. Okay, and particularly we want to look at the one that's going to be important in the ventral tegmental area. Okay, the one that's actually going to be present on dopaminergic neurons, and that's how nicotine is going to activate the dopaminergic neurons. We're going to have a nicotinic acetylcholine receptor on the dopaminergic neurons in the ventral tegmental area. Okay, and of course that's excitatory. Okay, if that gets activated, it will open and allow in sodium ions, okay, which will excite the neuron, okay, and nicotine is an agonist for some nicotinic acetylcholine receptors. But before I tell you about the nicotinic acetylcholine receptor actually present on the ventral tegmental area dopaminergic neurons, I firstly want to set the scene by telling you about the more famous nicotinic acetylcholine receptor. Specifically, I want to tell you about the one which is present on skeletal muscle cells. Okay, right. So, what we will do is look down on the nicotinic acetylcholine receptor from the outside, okay? So just like we drew for GABA A receptors, I'm going to draw a similar picture for nicotinic acetylcholine receptors. So we're looking down from above, and of course we'll have 5 separate subunits making up the full nicotinic acetylcholine receptor here. Okay, right. Now, the skeletal muscle nicotinic acetylcholine receptor is made up of 2 alpha-1 subunits, okay, 1 beta-1 subunit, a delta subunit, and an epsilon subunit. And let me show you how they're going to be arranged. So we're going to have the first alpha-1 subunit here, and we're going to have the second alpha-1 subunit here. Okay, so I'll firstly colour those in. So here is our here are our 2 alpha-1 subunits here. Okay, so those are those ones. And then we're going to have our beta-1 subunit, okay, positioned here. And remember, we are looking from the extracellular side, okay. So here in orange is our beta-1 subunit. Okay, next we'll put in the delta subunit. So the delta subunit is going to go here. Okay, so this subunit here. Okay, whoops, in red. Okay, and then finally, we're going to have the epsilon subunit here. Okay, in turquoise. Now, there is a subtle point here, okay, which could confuse you if you just type in skeletal muscle nicotinic acetylcholine receptor on Google. Okay, this is the one that is found on adult skeletal muscle. Okay, so on the skeletal muscle of you and I. Okay, adult skeletal muscle. There is a different one that is used on fetal skeletal muscle. Okay, now it's only subtly different. Okay, in fetal skeletal muscle, they use the gamma subunit here where the epsilon subunit is. So instead of having the epsilon there, you would have a gamma subunit there, and that's in fetal skeletal muscle. Okay, so you have a different one when you're in the womb compared to when you're actually an adult human. Okay, right. And if you Google it, if you Google nicotinic acetylcholine receptor on skeletal muscle, I can't guarantee which of these two you will see. Okay, so don't let that confuse you. Right. So let me now show you where the acetylcholine binding sites are then on the skeletal muscle nicotinic acetylcholine receptor. So the acetylcholine binding sites then, you have only two of them, again just like the GABA-A receptors, you only have two on this type of nicotinic acetylcholine receptor. Okay, now one of them is located between the alpha-1 and the delta subunit, and the other is located between the alpha-1 and the epsilon subunit there. Okay, so these are the acetylcholine, which I'll abbreviate to ACH, binding sites. Okay, so when acetylcholine is released by the presynaptic neuron into the synaptic cleft, what will happen is it will diffuse across, okay, and it will bind to these acetylcholine binding sites, okay, on the nicotinic acetylcholine receptor, and the nicotinic acetylcholine receptor will then open and the pore will become much wider, and it will then allow sodium ions from the extracellular fluid to come into the cell, which will cause excitation of that postsynaptic cell, whether it's a skeletal muscle cell or some other type of cell. Okay, right. Now let me show you the form that's going to be extremely important on the dopaminergic neurons within the ventral tegmental area. So the one in the dopaminergic neurons of the ventral tegmental area is actually much simpler. It's what's known as the alpha-7 homo pentamer. Okay, now what does this mean? Well, homo means the same. Pentamer refers to the fact that it's a 5-membered structure. Okay, so the alpha-7 homopentamer is going to be a nicotinic acetylcholine receptor constructed from 5 of the same nicotinic acetylcholine receptor subunits. Okay, and the 5 nicotinic acetylcholine receptor subunits are all going to be alpha-7 nicotinic acetylcholine receptor subunits. Okay, so if this is the receptor viewed from above, every single one of these subunits is going to be an alpha-7 subunit. Okay, right, and this is the form of the nicotinic acetylcholine receptor, or the form of nicotinic acetylcholine receptor that we think is very important in the ventral tegmental area. Okay, so I'll colour all of these alpha-7 subunits in blue here. Right, okay, now this is very different from this skeletal muscle one. Okay, this skeletal muscle one was what was known as a heteropentamer. Okay, it was made up of 4 different types of nicotinic acetylcholine receptor subunit. Okay, so it would have been known most definitely as a heteropentamer. Hetero means different. Okay, so this is a pentamer where the subunits that make it up are different. Okay, and it only had 2 binding sites for acetylcholine. The alpha-7 homopentamer has 5 separate subunits for has 5 separate binding sites for acetylcholine. Okay, so to open this receptor, you need more than you need multiple acetylcholine molecules to bind. I'm not completely sure as to whether you need all 5 to bind. I would suspect the answer is no. Okay, but you certainly have 5 acetylcholine binding sites. Okay, right. So let's now then talk about nicotine itself. Okay, actually before I talk about nicotine, let me talk about the fact that this will be on those dopaminergic neurons. Okay, so if we have our dopaminergic neuron here, okay, in the ventral tegmental area, many of the dendritic spines will contain alpha-7 homopentamer onto nicotinic acetylcholine receptors on their membranes. Okay, so if you activate these alpha-7 homopentamers on these dendritic spines, they will open and they'll allow in sodium ions, which will make it more likely that the dopaminergic neuron is going to fire an action potential from the axon hillock here. Okay, right. So this is how nicotine is going to make these dopaminergic neurons of the ventral tegmental area fire more often, by activating the alpha-7 homopentameric nicotinic acetylcholine receptors on the dendritic spines of these dopaminergic ventral tegmental area neurons. Okay, right. So, nicotine then, the active ingredient in tobacco, okay, well, as far as the addictive properties of tobacco are concerned, is an agonist at certain nicotinic acetylcholine receptors, hence why they are called nicotinic acetylcholine receptors. So nicotine will bind to these acetylcholine binding sites, and it will cause the receptor to open just as acetylcholine would, okay? That will then allow sodium ions into the cell and will make the dopaminergic neuron more likely to fire an action potential, and hence you're going to increase the rate at which these dopaminergic neurons will fire. More dopamine will get released into the nucleus accumbens and medial prefrontal cortex. Okay, now, one little point that I would like to stress is that nicotine is not an agonist at all nicotinic acetylcholine receptors. Specifically, thank God, it is not an agonist at the skeletal muscle form of nicotinic acetylcholine receptor. This form is all over your skeletal muscle cells, okay, and when it's activated by acetylcholine, it allows sodium ions into the skeletal muscle cells, and that causes the skeletal muscle cells to contract through a lot of other steps as well, of course. It excites the skeletal muscle cell, which then leads to the skeletal muscle cell contracting. Okay, if nicotine was an agonist at this form of nicotinic acetylcholine receptor, okay, then every time you smoked a cigarette, it would lead to involuntary unstoppable muscle contractions, okay, and it would most likely be fatal because it would cause problems with your breathing. Okay, so nicotine is not an agonist at all nicotinic acetylcholine receptors, but it is an agonist at the alpha-7 homo pentamer, which is the form that we think is important on the dopaminergic neurons of the ventral tegmental area, and this is how nicotine causes an increase in the rate of firing of these dopaminergic neurons in the ventral tegmental area every time you take it, and that's how it leads to addiction. Okay, right. So just before we go on to the opiates, which we'll do in the next video, I just want to say a few words about ethanol, okay, which is more commonly just known as alcohol, okay. So ethanol is addictive, and it does increase dopaminergic neurotransmission in the nucleus accumbens and the medial prefrontal cortex, just like all the other addictive drugs, okay, but the mechanism of action is not known, okay. It's a small molecule. We know very little about how ethanol achieves any of its functions because of the fact that it is a very, very small molecule and therefore does a huge number of different things, okay, but we do know that it does increase dopaminergic neurotransmission in the nucleus accumbens and the medial prefrontal cortex, just like all the other addictive drugs. Okay, right, so we will call it there for this video, and then in the next video what we'll do is look at the opiates.